All right, so everyone's heard me talk enough this year, so I'm going to hand it off to Dr. Bartel, who's going to introduce our wonderful speaker today.
Good news. We have good
Oh, there's good news. Yeah, but I'm going to do it next week.
I'm wondering what this good news is, but thank you all. And also thank you, Noah, for giving me this opportunity to introduce the speaker. It's a great day to have a speaker in, but it's also a special day, an auspicious day. It's the birthday of one Professor Noah Snyder-Mackler. And I was talking to Professor Martin earlier today, and he said that there would be nothing he would enjoy more than singing you the birthday song with the entirety of the faculty. 1, 2, 3. Birthday to you. Happy birthday to you. Happy birthday, dear Noah Snyder-Mackler. Happy birthday to you. Thank you all for your patience on that one. All right, it gives me great pleasure to introduce Martin Picard to ASU. He has an illustrious career, truly an expert in his field, I would say. He has a long history of work with mitochondria, originally getting his bachelor's degree at McGill in neuroimmunology and continuing on with his PhD, looking at the mitochondrial biology of aging in multiple tissues, but largely in skeletal muscle, was your thesis. So a wide range of views on the mitochondria. He then moved to UPenn for the Center of Mitochondrial and Epigenomic Medicine, where he did his postdoctoral work with Doug Wallace, looking at intermitochondrial interactions and reprogramming of the nuclear epigenome. So really focusing in on that topic. And he even had the opportunity to collaborate with the great Bruce McEwen from Rockefeller, probably the grandfather of stress in the brain. So really amazing training opportunities he's able to pursue. He then joined Columbia in 2015 as part of a translational neuroscience initiative where he has a appointment through the Department of Neurology and Psychology. Since then, he's won many illustrious awards, including the Herbert Irving Scholar Award from 2017 to 2020 as a fellow. He won the Neil E. Miller New Investigator Award in 2019, and most recently earned the Early Faculty Impact Award from the Federal Association of Behavioral and Brain Sciences. He's produced over 100 publications with over 8,000 citations. One of his most recent in eLife is a map of graying hair and stress, something many of us can relate to. And I hope you check out his recent submission to bioRxiv, which I suspect we'll get some previews of today. And so without further ado, I'd like to introduce Martin Picard, everybody. Thank you.
Thank you. Does this sound good? Yes? Clear? Thank you so much for the introduction. Thank you for the invitation. First time in Arizona. I love it. The weather is beautiful. All of the trees and the plants you have here is really special. What's that? All year long, yes. Many of you have offices packed with plants, and so there's a lot of interesting life here. So I've been enjoying this a lot. And also, Noah asked me to talk about mitochondria, which is my favorite topic. So we'll talk about energy and mitochondria and the mind-mitochondria connection that we're very excited about. So I'll speak about two topics today. First, we'll talk about energy and why energy is so fundamental to life and to the human experience and everything that we care about. And then I'll talk about mitochondrial phenotypes, mostly highlighting that there are different kinds of mitochondria. And I think most people here have probably heard about mitochondria at some point. So I'll give you a bit of a different story about them. And then in the second part, I'll focus on the brain. I know a lot of you are interested in that particular organ, and in understanding how that relates to behaviors and to our experiences. So why focus on energy and why focus on mitochondria? So if we look at this picture, this is kind of a general image, is there a pointer? I'll use my mouse, okay. So this is a general illustration of the different facets of human life, right? There's the brain that functions and produces cognition. It's based on, oops, I'll just go back. It's based on neural activity, and the organism mobilizes multi-system stress responses in response to the exposures in our daily life. There's cellular level processes like cell proliferation that keep your stem cells and your immune cells going. Immune regulation, wound healing, right? The ability to heal a wound, right, after you get hurt is energy dependent. And the whole process of development and aging also requires energy. So every little aspect of the human experience requires the input of energy into the system. Without energy, we would be a blob of cells, and then we would just follow the second law of thermodynamics and just decay, right? Entropy would win over. The fundamental ability of living systems that's unique to living systems is that we can kind of defy the entropic decay, and then by fluxing energy through the system, we can stay alive and grow and so on. So where does energy come from? Well, it comes from this really beautiful and complex process that we won't go into too much detail, but it happens inside the mitochondria, right? Every one of your cell is populated by hundreds or thousands of mitochondria, and they roughly look like this. They have this outer membrane and this inner membrane, and then inside, if we zoom in here, this is the inner mitochondrial membrane, and these proteins is called the electron transport chain, the ETC, or also the respiratory chain. Or the oxidative phosphorylation system, OXPHOS. And what happens here is carbon substrates that we eat enter into the TCA cycle, beta oxidation for fat, and then the electrons are ripped off from this. They're transported and given into complex 1 and complex 2 of the electron transport chain. Then the electron jump onto complex 3 and then complex 4, and complex 4 is where the magic happens. The purpose of breathing, and you can go so far as saying the reason we have lungs and we have a cardiovascular system and blood vessels and red blood cells is to bring oxygen down to your cells. Then inside the cell, oxygen goes to mitochondria, and then inside the mitochondria goes to complex 4. And this is the key to life. Oxygen accepts the electron that's flowing through the electron transport chain and then turns into water, which is what you see here. This provides a driving force. The whole driving force for life is oxygen taking the electron, and that energy fluxing through the chain is responsible for pumping protons across the inner mitochondrial membrane. So this then electrochemical gradient is what's used to, it's like a little battery, right? And then this is like electricity. You take chemical energy, you transform it into electrical energy, right? And then as we know, electricity can power your computer, it can power your phone, your car, your toaster, your hair dryer, everything. Electricity is kind of this versatile form of energy. So that's what happens in mitochondria. Maybe that's one reason why mitochondria were so important in allowing complex multicellular life, because it's this flexible form of energy. Unlike ATP, which is one thing that can do, you need to have an ATP pocket to use the energy. So where does the carbon substrate come from? Well, it comes from the plants, right? So the plants make up the food substrate that we eat and the oxygen that we breathe. And then in return, the byproduct of this in the mitochondria is water, like I said, and carbon dioxide, which is, interestingly, exactly what the plants need, right, to close this life cycle. And then where does that energy come from? Well, it comes from the sun. There's this, amazing quantum electrodynamic process of nuclear fusion that happens in this reactor in outer space, and then that energy, beams down, is captured by the plants, made into food substrates, chemical energy. We eat that. Mitochondria transform this into electrical energy, transforms this into energy that powers our lives. So energy is this key step and key factor in life. So life is a regulated energetic cascade, right, from the sun down to your mitochondria and into your mind, where harvested energy is used to perform work. And that happens inside interconnected biological systems. Here's an example of an interconnected biological system, right? This is from Homo sapiens, for those of you who don't see human brains very often. Those little spaghettis are actually white matter tracks, and this is the connectivity inside the brain. And the brain does two things that are particularly unique. It uses a lot of energy, right? To keep the brain running and to keep consciousness on, the brain uses about 1/4 to 1/5 of the total energy of the whole body. And by weight, the brain is much smaller, but so the energy density or flux of the brain is very high. And then there's constant energy flux. Even when you stop thinking, or if you try to stop thinking, you try to meditate, or you go to sleep, right? Then you lose consciousness momentarily for a few hours. The brain still keeps going at high rate and consumes a lot of energy. And also the mitochondria in the brain, as I'll talk about, are actually a little special. So there's something unique at the energetic level in the brain. But it's also full of connections, which is actually where the magic happens, arguably. There's emergent properties. Cognition is the emergent properties of interconnected brain areas. And now I think neuroscientists move past the single brain region does this. No, it's the interconnection, the communication between the brain regions that actually lead to cognitive faculties. So it's one of the most densely connected organ in the brain. It has longer range connection, and there's plasticity. So the brain is a network, right? There's an interconnected system, but the rest of the organism is also an interconnected system, right? And the body is actually a cell collective. Every cell needs to be aware of what's happening in other cells, and every cell is bound to every other cell through the circulation and through the flux of energy. So the major pathway for energy transformation is the mitochondrial network. And I'll say a few words about how we think about this. And mostly we think about the mitochondria as a mitochondrial information processing system, or the MIPS. So what do mitochondria look like? When you want to know what something does in biology, very often knowing what it looks like is actually informative, right? Form informs function. So there's a lot of young faces here in the audience. So you're a grad student, you're in front of your computer, and you want to know what something looks like. What do? You Google it, exactly. So you ask Google, what do mitochondria look like? And then Google tells you, well, it's this bean-shaped thing. There's this maybe swimming pool-like thing in the middle, and there's mitochondrial DNA in there. There's two membranes and everything. But if you actually have the chance to work in a lab with a microscope, and then you can put a living cell under the microscope, make the mitochondria fluorescent, and then you ask, what do mitochondria look like? You peek down the eyepiece. This is what mitochondria look like. So every colored structure here is a mitochondrion. And the color scheme is blue is closer to the cover slip and closer to the objective. Red is higher up in the cell, so it encodes the z-axis. But what you can see mostly is that they're all very diverse. There are different types of mitochondria, longer, shorter. They interact with one another. We can focus on this example here. These two mitochondria initially don't interact, but they start, and then this third one comes, then they share information, then this third one says, okay, I've had enough, and then leaves. So there's this whole social life, and there's actually, if you boil down principles of mitochondrial behavior inside the cell, there's a lot of similarities with social behaviors that you see among animals and insects. I won't talk about this today, but we've written about this if some of you are interested. So what do mitochondria look like inside the living body? Right? So we did a study to look at this in neurons, and specifically we looked at the hippocampus in mice. Then this is a Golgi stain. If you zoom in, you see the cell body and the dendrites, and there's the axon. So we looked at mitochondria in different parts of the cell. Right? And it turns out there are different kinds of mitochondria with very different shapes in different parts of the cell. So let's go inside a dendrite. A dendrite of the thing that's from a pyramidal cell in the hippocampus. So this is the dendrite. We'll zoom in, and then you'll see different kinds of mitochondria. Here's a little pink one. It's not too long. Then there's this brown one, this green one, and then look at this long brown one. So there's a single mitochondrion. It's over 10 microns in length, right? Which is very long. Actually, this one is probably closer to 20 microns in length, right? So it's extremely long. This is here the cell body of a cell body similar to where this dendrite was coming from. So this is the cell membrane in pink. It's going to come out. The nucleus here just disappeared. So now we're inside where the nucleus would be, right? And you're traveling among the mitochondrial network, right? And you see there's some long ones. You see the purple and the blue ones are kind of holding each other, and they're different shapes. Here, each color is a different organelle. And they're distributed all around the nucleus, which is relevant to what we'll talk about. So the mitochondria here all kind of encircling the nucleus, and they're all diverse. And this is, of course, a static image from electron microscopy, 3D EM. But if you look at this in real time, you see, like we saw in the video, that they're actually dynamic and move around. This is another example. That's a mouse embryonic fibroblast, a MEF, that you can put under the microscope. And then if you look at this and you start to ask a question we're very interested in asking, and others are interested, how do stuff, right? How do things from the outside world, right? Stressors, psychosocial stressors, right? Adversity and discrimination. How do those things trigger changes in the cell and then in the epigenome, right? And eventually in gene expression that modify aging trajectories, for example, right? And health processes and resilience. Well, if you look at this from this perspective, what we know is that this kind of black hole in the middle here is the genome, right? We're making the mitochondria fluorescent, and they're all encircling the nucleus. So that's where the epigenome, the beloved telomeres, and the demethylation process happens. This is where the environment is, right? Outside the cell. So the nucleus is encircled, right, physically, topologically, by this network of mitochondria. And if we ask, well, how do those things from the outside actually interact, right? What's the X factor that links the environment to the gene? And then we can ask, well, are the mitochondria this X factor, right? Are they this interface by which or through which, right, physically, how does a hormone actually make it from outside the cell through the cytoplasm inside the nucleus? It has to go through this mitochondrial network that's kind of powered by energy. And then maybe this leads in an emergent way somehow to health processes and dynamics. So that's a working hypothesis, right? That mitochondria is this dynamic interface, this energetic interface between the outside world and the inner world of the epigenome. So this is one way to illustrate this. Here we have every biological system needs to do three things, right? It needs to sense information, become aware of the surrounding world. It needs to integrate that information in a way that makes sense and in a way translate an exposure into something that the organism can recognize. And then third, that those first two steps are meaningful if the third step then can be to communicate that information, right? So you have a neuron, receives an input, integrates it, changes membrane potential, now fires or not, that's the output, to inform the rest of the brain, the rest of the circuit of what's happening. So mitochondria do this exact process in three ways. So the first is that mitochondria are sensitive to a number of inputs, and I won't go into the details of all of these inputs, but mitochondria are sensitive by having receptors, by having transporters to systemic inputs from the bloodstream, right? But also inputs from the cytoplasm and then from the cell membrane. And then in turn, mitochondria will then talk to each other, talk with other organelles like the lipid droplet, the endoplasmic reticulum, the peroxisome, and also with the nucleus, right? So that's the mito-nuclear interaction and the mito-mito interactions that integrate the information. So mitochondria will sense metabolic information, then convert that into changes in membrane potential, and then in turn produce outputs that the nucleus understands, right? So the mitochondria actually speak the language of the epigenome. And all of the cofactors and the substrates for epigenetic modifications, right, whether it's acetylation or DNA methylation, they're all made, all of those cofactors and substrates are all made in mitochondria or in a way that depends on mitochondrial metabolism. And to remove all of those modifications also involve mitochondrial metabolites. And then the third step is producing outputs, signaling outputs that will change what happens in the nucleus, but also will change what happens systemically. That's the release of a number of different outputs, either directly from mitochondria spitting something out into the circulation, or indirectly by turning on some genes in the nucleus, and then that turns on proteins that are released into the bloodstream. So that's what I meant by this mitochondrial information processing system, right? You can integrate information, receive or sense information, integrate that information, and then produce outputs that regulate organismal processes. This is this same idea here, but with a real cell. This is a picture of a human cell in culture. You see that the cell touches another cell here, another cell here. The nucleus is in yellow. And then each of the mitochondria here has the ability to sense information, integrate that, and then produce outputs. So kind of like your cell phone, right? Your cell phone will receive input, convert whatever cell signal into characters that you can read, your text, and then you can text back, and then there's an output that allows you to communicate with the rest of the world. So to the mitochondria and to the cell, right? Other cells, other mitochondria are the rest of the world. So mitochondria are the processor of the cell, not the powerhouse. And very importantly, in this whole ecosystem, there's this mito-nuclear interface. This is an example. This is a real picture, right? You have 3 mitochondria that are sitting just a few hundred nanometers away from the nuclear pores. This is the window, right? The window between the intracellular world of the epigenome and the genome, and then the outer world of making proteins and regulating process inside the cell. So we're starting to think about mitochondria as this not only interface, but also regulator of processes from organelle to organism. And mitochondria have this ability to talk to the nucleus and the rest of the body. So very important point, there are different types of mitochondria. And I had interactions with some of you this morning and no, and then said, oh, you should have a few slides about this. So I have 3 slides about specifically defining different mitochondria in different organs and tissue types. But first, I want to show these pictures. Mitochondria are different in different organs, right? So the 4 pictures show mitochondria in different tissue types or different organs. Any guess as to which tissue is this mitochondria from? Muscle, yes. Which kind of muscle? Cardiac, yes. Good guess. So you see the number of cristae? This is where the oxygen is used, right? The magic of respiration happens in those lines here, in the cristae. So and the heart consumes a lot of oxygen, so it has a lot of cristae. Any guess what this one is from? There's a big hint. It's an axon, yes, exactly. This is the myelin. Look at this beautiful myelin sheets. And this is a mitochondrion in the axon. And this is a mitochondrion from a non-myelinated axon that innervates the adrenal gland, right? The brain talks to the adrenal gland to trigger the release of stress hormones. So this is from a non-myelinated, this is myelinated. This one here, this is skeletal muscle, but it's sliced in the other direction, so you don't see the sarcomere. But look at this shape, right? This is not a bean, or this is not a peanut. So it has this beautiful kind of multiple arm, and you see the cristae here. It's very dense, right? And then this one is probably one of my favorite pictures of mitochondria. Which organ is this from? HP axis. Think about burrs. This is adrenal cortex. Zona fasciculata that produce cortisol. So the day life of this mitochondrion is not to make a ton of ATP, it's to make cortisol. And this is what the beautiful mouse zona fasciculata mitochondria look like. So very, very different, right? And in turn, actually all the steroid hormones, I didn't know this until just a few years ago, all the steroid hormones, including the beloved cortisol for those of you who do stress research, but also estrogen, progesterone, right, that actually make new life possible. You can't get pregnant if you're female without those hormones. Those hormones are made in mitochondria, in the ovaries. And testosterone. You won't get a beard and you won't get a penis during development if you don't have testosterone. Testosterone is made in mitochondria. So from an evolutionary perspective, why those hormones and the biosynthesis of those hormones happen to take place in mitochondria is unclear. We can speculate, but very interesting. And then mitochondria is also involved in the degradation of some other stress hormones, including the catecholamines. So how do we profile or how do we measure mitochondrial biology? I think I've tried to show how dynamic, how kind of alive the mitochondria are, right? How many things they do, and that they can do all of this because they're small living creatures, right? And we can profile living creatures at multiple different levels of complexity, right? On the left here we have DNA, so you can sequence a mitochondrial genome, right? But that doesn't really tell you much about what they're actually doing. Then you can look at RNA, their expression of that DNA. You can look at the protein that are encoded by those RNA. And then you can look at the activity of specific enzymes, like the complex 1 of the mitochondria. It tells you a little bit more what mitochondria are capable of, right? But not quite what they're actively doing. Then you can look at the activities of integrated system, like the ability to make ATP, which depends on multiple proteins. And then you can finally look at integrated mitochondrial behaviors and more complex processes. And here, I think from left to right, we increase the relevance of the mitochondrial health or the mitochondrial behavior to the end phenotype we're actually trying to model. The things on the left tend to be easier to measure and tend to be done more frequently because everyone can do PCR or look at mitochondrial DNA. But then the things on the right actually involve working with living mitochondria, which is harder to do technically. So one way that we've started to think about mitochondria is the same way we think about domains of human health, right? So you don't go to your doctor and your doctor says, okay, I'll study your function, right? We'll do a workout of your function. Well, cardiac function, hepatic function, or cognition, like which part, right? So we have all of these beautiful domains of human health that, of course, make sense to assess separately. Same thing for cell biology, right? You don't have a self-respecting cell biologist that says, I study cellular function. Well, which function are you studying? Yet we go around talking about mitochondrial function, right? And one point that I want to get across today is that there's no such thing as mitochondrial function or mitochondrial dysfunction, right? There are many functions and many ways to be dysfunctional. And there's all of these things here. These are different domains of mitochondrial biology that we can assess. And so there are different organ systems. We have systems in medicine. There are different cell types. Immunologists couldn't exist nowadays without a great classification system for different cell types. Same for neuroscientists. So are there mitotypes? Are there different types of mitochondria? So we started to probe mitochondria in different parts of the brain and in the body, and there's this beautiful data set called the GTEx data set, the gene tissue expression data set. 55 or 54 now different human tissues post-mortem, right? So people die and then they go and sample different tissues, do RNA-seq, and then those data sets are available. So this is what happens if you take only the mitochondrial proteins. So mitochondria have roughly 1,200 proteins that allow all of their beautiful behaviors. So if you take all of them and then you make a heat map, so these are all of the mitochondrial genes, and then on the top here are the 50 different tissues in this analysis. So you can see that this central nervous system, so all the brain tissues cluster together, so they have similar mitochondrial signature, right? We're looking at gene expression signatures for mitochondria. And then you can take all of this, because this is a little confusing, and then you can do a dimension reduction approach, right? You throw this into a PCA, a principal component analysis, or other dimension reduction method of your liking. This is what happens. So this is just with the mitochondrial genes, right? You can tell apart different tissues from other tissues. So all of the central nervous system tissue, these are all the brain, and there's a spinal cord in there. Here's a retina. So the retina is, it falls under the central nervous system, but it has different types of mitochondria. So the mitochondrial phenotype of central nervous system sits here. This is the mitochondrial phenotype of liver, which is pretty much exactly the opposite of the brain, right? It's regenerative and it's biosynthetic and all sorts of things that the brain doesn't do. So they have opposite mitochondrial phenotypes, which might actually be driving the ability of those different organs to regenerate or not. These are the muscles that have different mitotypes. And then these are glands, adrenal gland, parathyroid gland, which seem to have different mitotypes. And then these are all of the other tissues, digestive tissues and reproductive tissues. And you can go really deep into this. So I won't go deeper here. And you can do this at the single cell level, which is really beautiful when you start to look at immune cells and brain cells. And one major point here is you can also ask, how much do each tissue invest in making mitochondria? So this is an estimate of total mitochondrial gene expression that tells you how much of the transcriptome is devoted to mitochondria in each tissue. The heart is way up there. All of the brain areas are up here, and then you have other things here, including the white blood cells that are at the bottom, right? So there's a huge variation, spectrum, right, of how much mitochondria are expressed in different tissues. So mitochondria are different in different organ and the tissues and cell types. So we tried to look at this functionally, and this really matters if you're going to do studies about mitochondrial biology in humans, right? How do you actually measure mitochondrial biology in people? And then you want to ask questions about exposures and changes in mitochondria. Well, how do you do this? Well, you can sample blood, right? You won't sample the brain, but you can sample blood. Well, if the mitochondria are different in different immune cells, that's problematic, right? Or at least it's important to know. So we did flow cytometry to look at a number of different cell types, both from the myeloid and the lymphoid lineages and naive activated memory cells. So this is the design of that study. We recruited 21 people, 11 women, 10 men, that were fairly diverse. And then we sampled about 100 ml of blood, and then from most of the blood went into isolated white blood cells, and then with a 17-color antibody panel, we were able to sort on a 6-way sorter 8 different cell types. And then once you have the cell types, then you can profile the mitochondria, right? This is the electron transport chain or respiratory chain that I showed earlier, and then we can biochemically measure the activities of those different enzymes and look at the mitochondrial DNA and this marker of mitochondrial content, citrate synthase. So let's plot this. This is a 2-dimensional space for mitochondrial phenotypes or mitotypes in immune cells. And let's look at different immune cells. These are antibody-producing B cells, right? So you can see they have kind of medium citrate synthase activity, the x-axis here is how much mitochondria are there. Mitochondrial DNA copies per cell is the y-axis. So the B cells have the highest number of mitochondrial DNA copies, and nobody knows why, which is interesting. In comparison, these are the innate immune cells, neutrophils and natural killer cells, right? They're low on both. Very low copy number, 100 to 200 copies per cell, compared to almost 500 for B cells, and very low mitochondrial content. These are monocytes. They have the highest mitochondrial content or citrate synthase activity, but intermediate copy number. And these are naive or memory lymphocytes, CD4 and CD8. CD4 naive, CD8 naive. And then the memory cells in the same person, right, kind of migrate into this more common spaces. So the CD8 naive go here, and then the CD4 naive go here to memory. And then if you take total PBMCs, which is what it's easy and often done, and what we did for earlier mitochondrial studies, PBMCs are here. So they're nowhere like any of the immune cells that are in there. And that's most likely because they're contaminated with platelets. And platelets have lots of mitochondria and lots of mitochondrial DNA. So an important take home from this is different immune cells, right, and the humans that we study have different types of mitochondria, which is really important for measuring the effect of exposures on mitochondrial biology. And then looking at total mixed cells is probably misleading, which is a motivation to look at single cell data. So this is what we think we've been doing with mitochondrial biology. Has everyone seen this or something like this before, right? The blind scientist, like touching different parts of the elephant, right? And you see one that says, oh, that's a spear, and then, oh, it's a snake, it's a tree. Like, the thing is, every one of those is making a valid observation, right? They're making a valid observation about a part of the system. The thing is, they can't see the whole. And we think that's kind of what we've been doing with mitochondrial biology, right? You have a tool, let's say you measure ATP, or you can sequence a mitochondrial DNA. You make a valid observation about ATP production or about mitochondrial DNA sequence variation, but you're actually missing the integrative perspective. So we think that the integrative perspective is something like the mitochondria are a family of organelles, right, that exist as distinct mitochondrial phenotypes or mitotypes, defined by their molecular, morphological features, activities, functions, and behaviors. So this is a paper that's coming out this month that kind of lays out a framework to how to think about mitochondrial biology and how to move, our field forward beyond the function-dysfunction, simplistic model we have. And I just want to show this. This is modeled on Maslow's hierarchy of needs, right? Where you have like the basic stuff at the bottom, and then you get more complex things. So we can think of mitochondria in similar ways, right? There's like basic things like features, activities, and then eventually like the full realization of mitochondrial potential happens at the behavioral level. And very often we end up measuring just stuff at the bottom of the pyramid, and we don't really capture what the full complexity and beauty of mitochondria. So we've laid out some cell-dependent mitochondrial phenotypes, and I just wanted to show this, but not fully go into details. But the position of mitochondria, their shape, right, kind of falls into this. Then their mitochondrial features. This is the omics. That's where you tap, into with your omics, the proteomics, lipidomics, and you can tap into the static features. These are things that don't move and that don't change. So you can measure this in frozen tissue, and that's no problem. Then there are activities, right? Enzymatic activities, which get you closer to the actual dynamic nature of mitochondria, like membrane potential and protein synthesis. And then there are functions, like making ATP, which requires respiration, but also flow of electrons, and also this other protein, and this other activities. Calcium regulation and making steroid hormones, for example. And then behaviors, things that involve the whole mitochondria. Right? Mitochondria moving around, dividing, fusing, signaling, which are more complex and more difficult to measure, right? And more difficult to study. So the mitochondria are diverse and they're multifunctional. That's kind of the point here. So that opens up a lot of questions, and we're quite excited about this. So now let's apply this, and let's think about the brain. And I said mitochondria and the brain are a little different than other places in the body. And there are good reasons to believe because of what I mentioned earlier, right? The flow of energy through the system actually drives functions, and so perhaps the way mitochondria function in the brain actually drive behavior, right? And to what extent different, inter-individual behaviors are driven by differences in mitochondria. And when we bring people in the lab, we do stress studies, and we bring people in, and then we say, okay, now just relax. And we put a catheter in their arm, and we ask them a few questions, and then they just relax for half an hour. And then we tell them, okay, now you're going to have to give a speech, and you're going to be evaluated on your performance and judged. And then they have 2 minutes to prepare their speech, and then 3 minutes to deliver their speech in front of a camera, and a white coat wearing, very serious evaluator who just looks at them and says, you can start. And then they have to defend themselves against like a false accusation, and then the heart rate increases, and we measure blood pressure that it reaches like hypertensive levels, and we collect saliva in the blood, and cortisol shoots up, and it's this beautiful multi-system, integrated response of the whole organism, right? So that model, we didn't invent it. It's been used thousands of times. But it just allows you to see how the whole system responds, right, to a ecologically valid stressor that human and social creatures care about, right? How do other conspecific, or how are they judging me? So it's kind of it also taps into a very sensitive thing that humans have, which is this sense of self and the ego, and that's really potent stimulus for the HP axis and all of these other things. People respond differently. Right? One person, you subject them to this thing, and then they have a massive cardiovascular response. Blood pressure increases, heart rate goes to 160 beats per minute. And then another person, right, the next day, their blood pressure and heart rate increases a bit, but they release massive amount of cortisol. And then a third person, right, two systems kind of go up, but mostly they have this inflammatory response. Just as psychological stress is sufficient to trigger pro-inflammatory cytokine production, IL-6 and IL-1 beta, TNF alpha, just with, challenging the sense of self. Why? Why do these people respond differently? So we're after testing the hypothesis that those inter-individual differences are driven by mitochondrial differences, right? Or at least a portion of them. So that's kind of a guiding hypothesis for one of our NIH-funded clinical studies that we're running. So we asked that question first in mice, right? So how much of inter-individual differences in social and anxiety-related behaviors are driven by mitochondria? Because if you buy inbred C57 black 6 J or N mice, they're all genetically identical. Many of them have the same mom, right? They're from the same litter. They eat the same food. They grew up in the same environment. Yet, you test them, and some of them are quite anxious. Some of them are pretty chill. What's driving those differences? Could it be that there are differences in the mitochondria in their brains or elsewhere in the body that are actually driving those differences? So that was the question here. So Ayelet, a former student in the lab, got a cohort of about 40 mice, and then about a third was stated control, about a third was treated with chronic corticosterone in the drinking water, which is a well-used model to elicit depressive behavior, and then another third was treated with chronic social defeat for 10 days, which is a social model of chronic stress and also elicits depressive symptoms. So then we have whatever diversity existed that to begin with in the mouse cohort is now wider. There are some animals that are exposed to stress and some not. So now we have a very diverse cohort of animals, and then Ayelet went and cryosectioned the brain into multiple slices, and then each red dot here was a punch. So she went with a tissue punch, isolating specific anatomical areas of the mouse brain. So that was over 500 samples that Ayelet collected. That was the largest project we had done so far. And now we can profile the mitochondria in all of those little pieces of brain, right? And we can ask questions that haven't been asked before. Are the mitochondria different, or how are they different between different parts of the brain? How are they impacted by stress, which I won't talk about today? But then how do the mitochondria in those brain areas predict anxiety levels, right? Or social avoidance or social interactions. So these are the three groups that I mentioned: the chronic core to drinking water, the social defeat with the big CD1 aggressor mouse, and then behavioral tests, open field test, as well as three other ones. And then this is, for example, the percent of time that's spent in the center, right? So if you're a really chill mouse, you'll explore it. You don't really mind being in an open space. So these mice have low anxiety. These mice have high anxiety. This is actual data, right? This is a distribution of anxiety levels based on this test across the three cohorts. And so you can do this in the same animals as you do the mitochondrial profiling, right? And then you can look at how these correlate. So we measured 6 total mitochondrial functions or more activities. This was done before we, we finalized this other paper, across 17 brain regions. So then we end up with a big matrix where we have, we can plot the correlations. This is what the correlations look like. In this open field test, right, the more mitochondrial activity or function you have in this brain area, which is the primary motor area, the M1, the more anxiety the animals display. And this is here the % time in the open arm. This is the elevated plus maze, the mitochondrial health index, which is an index of mitochondrial energy production capacity. Again, the higher the MHI, the higher the anxiety. So this is the correlation matrix. These are the 4 anxiety-related tests. This is novelty suppressed feeding, and then social avoidance. And the more red the cell is, means that it's strong positive correlation, and the more blue it is means that there's a negative correlation. So you can see that different behaviors map differently onto the mitochondrial features. These are the 6 mitochondrial features that we measured across the 17 brain regions, and we also measured non-brain regions that are less associated. So coming back to this here, right? There's energy that matter and connection. So if we tweak the energy, or if there's this natural variation in the energetic properties or capacity of the mitochondria in the brains of these animals, does this relate to behavior? But we know that the brain with all these connections that makes more sense to think of the brain as a collection of networks and circuits. And if you look at brain activity, using fMRI, for example, you can find that some brain regions have fluctuations in activity over time. You can create a similarity matrix, which is based on brain regions that if this one goes up, this one also goes up, right? And then if this one goes down, the other also goes down. And you can map patterns of functional connectivity and then project this into a similarity matrix and then build functional brain networks. So that's things that people have been doing for a long time in fMRI. And then there's those networks exist at the scale of the whole brain and also at other scales. So we asked, could it be that the mitochondria in the brain are also organized as networks? So we have 17 different brain areas, 6 different mitochondrial measures for each. Is there evidence that they cluster in some way? If the mitochondria, null hypothesis here is that if all the mitochondria are the same, then we just have one big blob, right? If you have, let's say, 2 brain areas that have the same mitochondria, those will be connected, but they'll be disconnected from the rest of the brain. So let's look at mitochondrial connectivity in the brain. This is the similarity matrix of the mitochondrial features based on the mitochondrial activities. Here's one brain area, that's the cerebellum, and then the 6 different mitochondrial features, right? And then the nucleus accumbens and the 6 mitochondrial features. The hippocampal CA3 formation, the 6 mitochondrial features, and so on. And then they're correlated with all of the other ones. Right? Does that make sense? That correlation matrix? Red means they're positively correlated. If all the mitochondria in the brain were the same, this whole thing would be, deep red. What you see is that it's not deep red, right? And there's some brain areas that are correlated with each other. You see here this cluster of the medial prefrontal cortex, the caudal putamen, and the M1 primary motor. They tend to be, they form a cluster to be correlated to one another, but they're not correlated to these other regions, right? And then notice that the rest of the body, these are muscles, adrenal gland, liver, and the heart. They're not correlated with the brain. So I won't go into this, but the animals with the highest mitochondrial activities in the brain are not the animals with the highest mitochondrial activities in the muscle or in the rest of the body. So there's kind of a disconnect here, which is fascinating and leading to some other hypothesis. So other brain networks that share similar mitochondrial properties is the question that arises here. And we worked with Manish Sagar at Stanford, and he deployed his computational whiz. He's a mathematician, and then he was able to use the different mitochondrial features and then project this on a multi-slice community detection method. And basically what this shows is that there are mitochondrial networks. And this is the similarity here is highlighted by the length of those nodes. These are the brain regions. So there are these 5 brain areas that form network 1, and then the blue one and the purple one. And then we can ask, are the mitochondrial activities in these networks predictive of behavior? So that shows, and that's the first demonstration or a hint that mitochondria actually cluster functionally within the mouse brain? And the answer is yes, the mitochondria in the brain, specifically in network 1, which is this network here, actually predicts the anxiety based on the open field test or the elevated plus maze or the social avoidance score quite well. And this is the summary of the results, and we'll just focus on network 1 here. These are the R, right? How strongly or the mitochondrial activity is correlated to behavior. So if you square these numbers, if you square 0.7, you get a 0.49, right? So about the R square is about 0.5, which means that there's about up to 50% of the explainable variance in the differences between animals that's explained by the mitochondrial activities in the brain, which is astonishing, especially given that the behavior is really noisy. Everyone who's done mouse behavior knows that the same mouse won't behave the same from day to day. So we know there's a lot of noise there. And the mitochondrial measures are also noisy. So you combine those, there's not 100% of the variance left to explain. So 50% of whatever is left to explain is quite large. And then we looked at this with gene expression analysis, right? I showed the mitotypes earlier. So you can kind of do a similar thing, but based on gene expression. This is the Allen Brain Atlas. So completely different cohort of mouse, right? This is out of sample. Let's look at gene expression. Is there evidence that the gene expression signatures in those 17 brain areas actually map according to the networks? So what you can see here is a 3D PCA. So again, we're just using the mitochondrial genes. There's 946. And then we're projecting this into this kind of unbiased 3D space, and what we see is that the network 1 region all cluster together, right? So those brain areas here all share a similar mitochondrial phenotype based on the gene expression signature. And then you can do this at the level of mitochondrial pathway. So instead of looking at the genes, you can say, okay, out of the 946, there's 12 of those that are involved in calcium regulation, right? So then those 12 of the 946 become a single pathway or a single line on that map here. And then each column is a brain area. So what you can see here again is that those different brain areas, they cluster according to their network. So network 1, again, the brain areas cluster together. And the null hypothesis, if all the mitochondria were the same across the brain, is that each column would look the same. So the fact that each column is different shows that there are different mitochondrial phenotypes or mitotypes across different brain areas. And that's work that Anna Munzel in a postdoc in our lab is leading with Jack Devine. So are there brain mitochondrial phenotypes linked to psychosocial exposures in humans? And this is work that Caroline Trumpf, an assistant professor in our group, has tackled in a beautiful cohort in humans where we have data on self-reported measures for up to 25 years. And then when the person dies, then we get the brain. And then we get a piece of frozen brain. You can do proteomics, transcriptomics, single nucleus RNA-seq, and we have colleagues who have done these multi-omics in these brains. So Caroline asked, well, maybe there's an association between how people feel, right, and the psychosocial exposures, the environment that they live in, and the mitochondria in the dorsolateral prefrontal cortex. And this is what she found. This is the mitochondrial complex 1, right, where the electrons enter, the very important part of mitochondria, and on the x-axis is the combined psychosocial exposure, positive and negative. So the further you go on the right, the more favorable the experiences. And here that R square is 0.18. So there's apparently 18% of the variance, right? Some people have more complex 1 in their brain, some people have less complex 1 in their brain. 18% of those differences between people would be driven by the experiences that these people have before dying. And whether that's the mitochondria driving the self-report, right? You have more complex 1 in your brain and then you feel better and you feel more connected to others and you feel more gregarious, or whether it's actually the social environment that's affecting the mitochondria, we don't know, right? But there's this connection, and to our knowledge, this is the first documented connection between mitochondria, brain mitochondria, and the psychosocial experience. So in summary, this is kind of the mitocentric worldview. And what I've showed you is how the biology of mitochondria and energy, right, at that level influences how cells work. And the ability of cells to talk to each other is actually contingent upon them being energized and having normally functioning mitochondria. And then the ability of cells to talk to each other, right, is actually the foundation for brain circuits and brain function, cognition, and then allows organs to function. And then the interconnection between the organs through hormones and through mitokines and through energy flow is actually the basis for people to function well. And then the ability of people to talk to each other and do what we're doing now is actually what creates great communities and a better world. So there's information flow, right? Your energy flow from mitochondria to the cells, to the organs, to the people, and to communities. But there's also flow of information the other way around, right? And what we experience and the subjective human experiences actually trickle down to change the heart rate, right? And then to change how neurons are firing in your brain, and then eventually to change your mitochondria. So that's this bidirectional mind-mitochondria connection that seems to be emerging from some of the data I showed you and some other things. This is our amazing team. We're really fortunate to have a number of people who come from different kind of background. And Caroline, who I mentioned here, her background is clinical psychology. And she trained as a mitochondrial biologist, and now she's a mitochondrial psychobiologist. So we're now trying to train a few more mitochondrial psychobiologists. I mentioned Anna, she's doing a lot of single cell analysis now, the mitotype analysis with Jack. Ayelet did the mouse brain study with the behavior and the mitochondrial phenotyping together with Alex. These are our collaborators. There's work that I didn't show in the human brain that has involved a number of people. We've collaborated with Rebecca and Susan on the white blood cell and mitochondrial phenotyping. Phil de Jager is our collaborator at Columbia who does all of the multi-omic and the single cell analysis. The three-dimensional mitochondria I showed was done by Amy in Newcastle, and my colleague Orion Chirihai at UCLA, who's worked with me to develop this mitochondrial information processing system. So if there are questions, I'd love to hear your thoughts and know what you think. Thank you.
Got time for some questions?
Yes.
So it's beautiful, and really appreciated the depth right there in terms of making this argument on mitochondria being very different. And so it's hard not to think about whether this correlation has a positive element. And I was just curious, how far are we from taking mitochondria from one cell type, sticking them in another,
Yes.
and actually seeing if that changes their phenotype?
Yes. Yes. So yes, thank you. So how far are we from taking mitochondria from one cell type, putting them into a different one, micro-injecting them, right? Or and then seeing how this affects cellular phenotype in the recipient cell. This is a great question, and I think we're pretty close. And there's studies that are being done now and that have been done in animals where you extract mitochondria from one animal, then you inject them in the bloodstream of another mouse. And then you see that these mitochondria infiltrate or get incorporated by the heart and the brain, and so the different organs seem to be able to take circulating mitochondria. And there's work we're doing on cell-free mitochondria and cell-free mitochondrial DNA, where normal living people have mitochondria circulating in their blood. And I think the most likely purpose of those cell-free circulating mitochondria is kind of sustaining the mitochondrial collective, where you have some parts of the body that can release mitochondria and other parts can take them up, as needed. So I think that's a really cool question that would go a long way in testing the hypothesis. Yes?
So you were mentioning that there's different types of mitochondria in different organs, and you were talking about boredom. Do we think that there's and maybe you mentioned it, maybe I missed it, but does that change over a lifespan? Do you see differences in numbers and functions and, through development and then aging?
Yes, great question. So we're in the process of doing yes. Do mitochondria change, and specifically do different mitotypes and specific cell types change across the lifespan? The answer is we know that mitochondria change across the lifespan. They don't become dysfunctional, I think, in a simple way that is generally assumed. So we're in the process of testing the hypothesis in the brain and in immune cells in a cell type specific way in two different cohorts. So that hasn't been done in a cell type specific manner, as far as I know. Yeah. Yes, Cheryl.
Wonderful talk. Thank you. It's very informative. I'm curious. One thing I really appreciated was the different behavioral
Yes. So question is, the mito-behavioral correlations that I showed, did we look at whether it was different between females and males and whether that's related to cognition as well? We committed the sin of only studying male mice in this study. I apologize. Otherwise, it would have been over 1,000 samples, and we would have been underpowered for other things. So yes, if this was to be redone, we would probably do it differently. So we don't know about the sex differences. I suspect there would be differences because there's so many things about metabolism, energy metabolism, that's different between women and men, or between rodent females and males. We didn't look at cognition, and that would be interesting to look into. The behavioral test, this anxiety-related task, did not all show the same thing. The open field test, the elevated plus maze, which maybe tap into something pretty similar, did show converging, effects. The novelty suppressed feeding, it's kind of it seems like to involve a number of things, like is the animal hungry or not, and maybe there's like regulation of whole body energy metabolism there, and combined with like motivation and smell, and so that was kind of a discrepant, result. And then the social interaction or social avoidance was actually, correlated differently. So the animals with the highest mitochondrial activities in the network 1 were actually the most social, and they wanted to go see other animals, but they were also the most avoidant of this open space and the open arms. So there's something different there, and or kind of loose hypothesis is that the animals which have high mitochondrial activities in those brain areas maybe feel like they have a lot of energy, and if they were to get in the confrontation, they could handle it. And if you're kind of borderline and you have low mitochondrial activity, then maybe you're better to avoid getting into a potential conflict. I don't know if that makes any sense, but yeah, good question. Yes?
I kind of goes along with future directions, but is there any thought about looking at the mice now, but the dams and the offspring, and see if there's any similarities or differences that transfer, or again with the female mice versus that have not been bred versus ones that have been bred?
Yes, that's a great question. Are the mitochondrial phenotypes heritable? And the mito-behavior correlations, are these heritable? Thank you for elaborating on the question a little bit more. We don't know, and that's a really good question. We suspect that those variation, all these animals have the same mitochondrial DNA sequence. So it's not differences, variation in mitochondrial DNA polymorphisms like we all have in this room. So it's not genetically driven. So if it's not genetically driven, also what I showed is that there are different brain areas that don't correlate with one another, right? And the brain and the periphery also doesn't correlate in most animals. So the animal with the lowest mitochondrial activity in the brain can have the highest mitochondrial activity in the liver and in the heart. So there's not an animal-wide factor, right? It's not like a trait-level feature that is genetically encoded that drives how much mitochondria or how well your mitochondria can transform energy. So there's something else, and our best guess is that they're kind of individualized strategies, right? Each organism, each animal, each person, might find a way to be healthy and to maintain homeostasis and to reach kind of a good stable point through different strategies. And people have called this equifinality, right? So there are this trajectory, different combinations of factors, but you end up at the same point, right? Which is homeostasis or health. And maybe there's a limited amount of mitochondria that is available to the organism, and the organism can flux a limited amount of energy, and that's a concept in the human energetic literature. And if that was the case, then you would have to partition or to kind of distribute your mitochondrial resources maybe differently and across organs, and so and that ties in with the cell-free mitochondria. So it's there's lots to think about there. Good question. Yes.
Do you see any evidence that you're working with that the mitochondrial functions that you've evaluated diverge as organisms age? And can that explain some of the differences you saw in the first?
Yes. Do mitochondrial functions, thank you for putting an S there, diverge as the animal age? This is a really cool question. And if we go back to development, before aging, or maybe some people think when aging starts, all mitochondria in the body start from a single mitotype in the ovocyte, right? And then you get the mitochondria start to differentiate into different mitotypes, the same way that there are different cell types that differentiate from the stem cell population. So that definitely happens through development. Through aging, we don't really know. So we're going to have an opportunity to look at this in different immune cell types from single cell RNA-seq in people between ages 20 and 80 years old. So I suspect we'll see something interesting there, and I suspect different cell types are going to, different mitotypes and different cell types are going to behave differently, change differently over time. And we've done some in vitro studies. We developed a cellular lifespan system, so you can take human skin cells, put them in a dish, and then every week you monitor them for 9 months. So a whole human gestation period. Very courageous student who did this. Every 5 days you come in the lab, change a media, passage your cells. And then if you do this, you end up with a longitudinal study, right, with very high temporal density of metabolic features, so you can do metabolic assessment, gene expression, RNA-seq, the EPIC array, so we have longitudinal DNA methylation changes, and there's some just beautiful changes that match, the epigenetic clocks that were developed in human tissues. For those of you who know this, they work in vitro, right? Like you can track the aging of cells through the change in the PC clocks, especially, and the telomere shortening and gene expression signatures, GDF15, which is like an aging marker in humans. Actually, those cells, as they get older, they start to secrete more GDF15. So there's a number of aging human biomarkers that actually show up in this longitudinal cellular system. And we're just starting to look at the mitotypes from the RNA sequencing to see how that changes over time. So that hasn't been done, but it's in progress. Yes?
2 questions. Just because one, based off what you just said, and can you take it from an embryonic stem cell and differentiate them into different cell types and do the mitotyping?
Beautiful. Yes, that's part of the plan. Yes. Yeah. So can you do this? Can you track the changes in mitotype starting from a stem cell that you differentiate into different lineages, different cell types, and then how do the mitotypes change over time? And then you can ask questions if you have sufficient temporal resolution. Do the change in mitotype actually precede the differentiation into a cell type? Because there's a lot of really good evidence in the stem cell literature that the change in the mitochondrial phenotype actually is necessary for the differentiation into certain cell types. And like the polarization of macrophages from like M0 to M1 or M2, you can actually direct a cell differentiation by tweaking mitochondria, and you can prevent differentiation in some models from stem cells to a differentiated cell if you block fatty acid oxidation, so if you block different domains of mitochondrial biology. So that field is fascinating, and it shows that mitochondria play an instructive role in many aspects, fundamental aspects of cell biology.
I have a second question. Because it goes back to some of your earlier slides, and it might be just a graphical thing you use, but I'm kind of curious if it's true. You had some polarization of signaling processes in the nucleus with the mitochondria around, saying that there's this directional flow. It was like a rainbow, so is that actually the case? Like, are there certain proteins localized on one side of the mitochondria to relay information to the other side, or was it just for
this?
Yeah.
No, it's bidirectional. So this is for illustrative purposes. So the question is this flow information really unidirectional from mitochondria to the nucleus? So there's a few arguments that we detailed in this paper why the flow of information might be privileged from mitochondria to the nucleus. One of them is that the warmest compartment in the cell are mitochondria, right? And how does heat diffuse? From hottest to coldest, right? Hottest things are more jittery, and the coldest stuff is less jittery. So the heat always propagates from warmest to coldest. So if mitochondria are the hottest element in the system here, right, then that might kind of promote the diffusion of information away from mitochondria. So nobody's ever proven this, and I don't know how we would do that. But there's most likely a lot of information and metabolites that can diffuse away from mitochondria, and then the nucleus happens to be surrounded by different mitotypes. But then there's definitely information that leaves the nucleus to nuclear pores, and those little black things here, these are the ribosomes, right? And that's probably endoplasmic reticulum, these vesicular things. So this is where proteins are made, right? So the RNA that's transcribed here leaves through the pore and then goes and then becomes a protein here on the ribosome. So it's bidirectional. Yeah. Yes?
Have you already looked at or really plans to look at like these mitotypes in different kinds of cancers?
So have we looked at mitotypes in different types of cancers? We have not. But there are data sets out there and that we could do this in, or others could do that. And it's clear that mitochondria are weird in cancer cells. It's kind of a hallmark of, and some people think it's a necessary shift that needs to happen, the bioenergetic shift. It's another example where mitochondria and mitochondrial metabolism play an instructive role in either oncogenesis or maybe the removal of cancer cells. So we haven't looked at that, but I would suspect that would be one model where things would be pretty different. And we know that cancer cells mostly don't rely on their mitochondria for energy. And one, I think, very cool hypothesis in cancer is that cancer cells are cells that have kind of reverted to a more primitive state of existence, right? Like endosymbiosis came 1.5 billion years ago, and then that allowed cells somehow to become multicellular. And our hypothesis at this point is that mitochondria actually allowed cells to become more social, and to actually, it increased their ability to compute information, right? If the mitochondria is the processor of the cell. So basically mitochondria acquired a sophisticated chip processor by acquiring mitochondria. Now all of a sudden they could talk to other cells and then build multicellular bodies and everything. And in many ways, cancer cells have kind of ditched their mitochondria for energy production, and now they start to use mitochondria for biosynthetic material to make lipids and building blocks to replicate. But they're kind of disconnected from their mitochondrial metabolism the way that normal cells are. So maybe mitochondria play some role in keeping cells in tune with the rest of the cell collective, which at this point is complete conjecture. Yeah.
Awesome. Let's thank Martin for a wonderful seminar.
Thank you.